Think of a deck of cards, a mix of images that repeat and can be used for a game. Now, think of DNA sequencing. The two don't seem like they would go together, correct? But there are many ways to create a metaphor. For today, we'll be exploring Sanger's method of DNA sequencing through our deck of cards.
In 1977, Frederick Sanger came up with a way to describe DNA sequencing which involved the use of chain remnant inhibitors. He wanted to find out the sequence of nucleotides in a single strand of DNA. Knowing the sequence of DNA in an organism's genome can assist biologists in understanding genetic diseases, agricultural scientists to breed or modify crops, and even identifying criminals in forensic sciences.
First, some background information on DNA strands and sequencing. To start, DNA is made up of a chain of 4 nucleotides. These are adenine, thymine, guanine, and cytosine. These nucleotides are called DNTPs, or deoxyribonucleoside triphosphates. In regular replication, to make a double strand of DNA, DNA polymerase adds the complementary base pairs to each nucleotide, DNTP, and creates what we know as the double helical structure, with adenine to thymine, guanine to cytosine, and vice versa. Polymerase acts as the enzyme that adds these new nucleotides to a lengthening DNA strand by catalyzing a chemical reaction.
In the metaphor's case, aka our deck of cards, think of the 4 card suites: hearts, diamonds, clubs, and spades, as the 4 nucleobases, aka our DNTPs. In this case, adenine is going to be represented by the black spades, guanine by black clubs, thymine represented by red diamonds, and cytosines by red hearts. To replicate the idea of complementary pairing, let's keep in mind that the spade and clubs are black, whilst the hearts and diamonds are red. Let's say that red is the color for our double-ringed bases, our purines, and black is the color for our single-ringed bases, our pyrimidines. This makes for a better visual for complementary pairing. Red goes to black, black goes to red. For the enzyme polymerase, well, it acts as the metaphorical glue in this case. But how does that even occur regularly without all the Sanger sequencing mystery? That part we have not told you yet.
Regular